So this last slide is piglet duodenum, and the duodenum is the very first part of the small intestine. So straight away after the stomach, the first part of the small intestine is the duodenum. On this slide again we have cells stained pink, nuclei stained purple, but also we have an added stain which stains mucus bright blue. And the sort of things that we're going to see here is more smooth muscle, so more involuntary muscle that you don't have control over. We're going to see glandular tissue, we're going to see villi, which are finger-like projections, and mucus secreting cells. Now, so here's a textbook of an intestinal tract, and we're looking at the very first part of the small intestine as it exits the stomach. And the structures that you see, these are textbook representations. So up here is the whole intestine. Inside it's folded and is covered in these villi, these projections. And this is all to increase the surface area. This is a very small tube, but this is the major site of absorption of all the nutrients that you eat. So not only is this a site of digestion, so where you break the material down, but also absorb it. So you can get by without a lot of your stomach, a lot of your large intestine, but the small intestine is very important. So to get the big surface area that is required to absorb all these nutrients, you have all these projections. So under low power, this is what that small intestine looks like if you take a slice through it. So this is a tube, but it's not a tube as we saw in the airway. There's no nice big space down the middle of it. And that is because this is a very soft piece of tissue. If we look at the real thing, we'll see that there's very little structure to this tissue. So the small intestine, while still being a tube, when you actually look at it, doesn't really look like a tube. And that's because it doesn't have the structure that something like maybe the trachea here has. This has got cartilage rings around it and holds it open in a tube, a bit like your tube from a vacuum cleaner. This here is the small intestine from a sheep, and you can see it really doesn't have any structure or rigidity, and actually it's very difficult to find the hole down the middle of it. I mean, I could stick my pencil down there, but there is no open hole down the middle of the small intestine. When the food goes down, it's pushed down by the muscle there around the outside, but there's no continuous opening, if you like. So both tubes, but quite different structures. So it's very much more like a tube as in the, say, the arm of your sweater. If you take your arm out, the space down the middle disappears. So this folded part here is the center of the intestine, and the food is pushed through. There's no big open space for it to fall down. Now, looking at it under low power like this, and this is actually about 4 images joined together. We couldn't actually see the whole thing in one screen. You can make out pretty much 3 different regions. On the outside here, there's a nice pink layer. Sort of looks a bit like that layer that was around the airway in the lung, and that's because it's the same material. You've got smooth muscle around the outside here. then you've got a very folded layer which is a sort of a paler pink colour. This is all glandular tissue. Glands basically secrete material, so this, the cells in this glandular layer are manufacturing material and secreting it into the centre of the small intestine. And then there's another layer here which has got this sort of blue colour in it, all folded up, and is actually those finger-like projections which make up the inside of the small intestine. So if we look at them under a little more magnification, we'll learn a bit more about them. So this is the very outside of the small intestine, and you can now see that that pink outer layer is actually made up of 3 different bands. On the very outside is a very thin layer, and that is something that you come across commonly every day. That is the serosa. It's the sort of skin around the outside of the small intestine, and that's what sausages are packaged in. So all of the material is stripped away from inside to just leave this skin, and that's what becomes a sausage skin. So that's that very thin outer layer. Then we've got 2 bands of pink. One, the outer one looking quite grainy, the inner one looking quite smooth. These are both layers of smooth muscle, so the grainy appearance is because it's been cut across. So the outer muscle layer on the small intestine runs along the intestine and it's been cut across. So you imagine a tube, it's running along it, you're looking at the ends of those muscle fibres. The inner layer wraps around the small intestine, and those 2 layers working together move the food through the small intestine. It's a process called peristalsis, and it's a combination of this inner layer contracting and squeezing the small intestine, and the contractions run along and push the food in front of it. That, as well as the outer layer contracting and sort of lengthening and shortening the small intestine, those 2 things working together push the food through the digestive tract. And then inside here we have this glandular layer, and you can see little rings of cells around little gaps, sort of an open space. So those are cells that are manufacturing, in this case they'll be manufacturing digestive enzymes. They secrete them into the centre of that little ring, and that will be connected up into the centre of the small intestine. So those digestive enzymes manufactured here, secreted, and end up in the lumen of the small intestine. And it's those enzymes which break down the nutrients that have come out of the stomach. Up until then, the digestion has been mechanical with the teeth and the mixing of the stomach, mixing with acid, but here we get enzyme digestion, which breaks up those complex molecules into simple molecules so you can absorb them. And moving to the middle of the structure, so here's the center where the food will come through. You can see the end of the glandular layer here, and these are these long finger-like projections. So the digestive enzymes are secreted from here into here. They break down the complex molecules that are in the lumen of the small intestine, and then these cells that line the villi absorb those nutrients, transport them through the cells into the middle of the villi, and they get carried away in the circulation. The other thing you can see here is bits of blue. So there are certainly towards the bottom of the villi you can see blue, and that's the mucus. And if we go to higher power, so here are these villi projections, and you can see some cells staining blue. So they're the ones that are secreting mucus. The mucus basically just helps the passage of the material through the small intestine. The other thing that it does, it also gives some protection to the villi because the material that's come out of the stomach is acidic and has to be neutralized. So you get bicarbonate buffers coming from the pancreas into the small intestine to neutralize that acidity. The other thing I should say about these villi is that these cells here are working very hard and they don't last very long. The tips of the villi are continually falling off. So a lot of the animal's feces is actually its own cells, and that is these cells from the villi. So the cells are being renewed at the bottom, but gradually they'll move up the villi and fall off. So in a later lab you'll learn about this endogenous nutrients that you find in feces. When you're trying to work out dietary requirements for animals, you have to take into account that a lot of the feces are actually part of the animal itself.